Or you could just check next to you and choose something.
Hey,
How are you?
I'm good.
Yeah, you're ready for this, right?
Yeah, well, we'll see how it goes.
Okay, okay.
I discovered while practicing that I had COVID over between Christmas and New Year's.
Uh-huh.
I have asthma too, so
Oh, I'm sorry
we'll
about
see
that.
we'll see if I can make it through an entire hour without like having to stop and be like,
Yeah.
all right, I promise I haven't run a marathon.
Okay,
Unexpected consequences, I guess.
I know. So it's just, that's what it is. And you have a side effect after you
Yeah.
get a COVID, and you just need to get it over for a little bit. Yeah, that's what I heard for most of the people.
Yeah, my pulmonologist said that he sees people that still like 6 months later
Yeah.
are finding things that they were used to do perfectly fine and are still
Mhm.
having a little bit of.
Yeah.
I'm like, I've been vaccinated, boosted, and then I have it. I've got like super immunity.
Yeah, I'm glad so you recovered quickly. Yeah. Yeah.
Yeah,
Yeah.
my husband gave it to me. He was over it in like 2 days.
Yeah.
Yeah, that's the way it goes.
Yeah, so I think it will get better. So because getting warm and so summer should be good, but who knows whether it come back. There's no winter. So
Yeah, well,
Yeah.
Baltimore's weather has been crazy. We had the weekend before, it was almost 80 degrees on Saturday, and Sunday it snowed,
Yeah.
and we had everything
oh
covered
my
in ice.
goodness, yeah,
Yeah, it was great.
yeah,
I'm like, can we just pick a season, just
Yeah.
one,
yeah.
or at least maybe bordering seasons?
Okay, yeah.
Say it to you. Sometimes people register and other times. I appreciate it.
Great, everyone. I think we're going to get started. My name is Marie Hurley, and I am the NGS Business Development and Product Specialist for Genomics at Agilent. I think everybody noticed that there are sandwiches in the back of the room, and there are also bottles of water. I really do not want to carry those bottles of water back across the parking lot, so please take a couple on your way out. Stay hydrated for the rest of the day. And before I actually get into the talk, I will apologize in advance. My daughter is having oral surgery in Baltimore today. It was rescheduled. So if I glance at my watch, it's just to make sure that she's doing all right. And with all of that being said, thank you guys for coming today. Thank you very much for having me. I don't think it is any surprise that the last 2 years and in a pandemic situation has changed how everyone has been doing business. Large gatherings like conferences and seminars have been moved to virtual or they've been canceled altogether. Face-to-face meetings have been few and far between. And all of that means that major product updates, announcements, have the potential to go unnoticed. Again, I can't tell you how happy I am to be here in person, and I wanted to share with you guys how Agilent is moving genomics into 2022 and beyond. There have been some pretty big updates that have happened in the last 2 years, not only with how we produce our products, what products we actually offer, but how our products are being used in the genomic space. Just a tiny bit of background about myself. I am from Baltimore, Maryland. I live actually a little bit north and east of the city. I was a lab manager at a high throughput genotyping and sequencing facility at Hopkins for about 15 years. I got my master's degree and moved to this role 2 days before Agilent, or 2 days before Maryland went into lockdown for the pandemic. So I picked the best time ever to do a career change. But it's been really good actually getting out to see and seeing customers instead of doing virtual meetings. So again, thank you guys all for being here. Come on. There we go. So genomics itself is a pretty broad category, and the products that Agilent offers in this space are no different. This slide is kind of encompassing of all of the products that we offer in the genomic space. Today, I'm going to focus on our NGS workflow and some of our synthetic biology products, specifically our single guides and our oligonucleotide libraries. If anything on this slide seems like something that you would like more information about, you can either let me know after this talk and I can put you in touch with the account manager, or this is my plug for the day, tomorrow at 11 o'clock in this room, there is actually a vendor show, which your account manager, Tin, and I will also be at. So, feel free to stop in, see the vendor show, get another lunch. So I'm gonna start with our next generation sequencing. And we have definitely seen some major updates with our Shore Select chemistries, how we create our baits and how those products are being used for applications like HiC and off-target analysis for synthetic biology. So the past several years has seen Agilent kind of switch from not only just being a product vendor, but to be more of a workflow or a solutions vendor. Agilent offers solutions for most of the NGS workflow, the exception being some nucleic acid extraction and the sequencing part. But we can seamlessly plug those exceptions into our workflow. We offer solutions for QC with our automated electrophoresis portfolio, library prep options, target enrichment, and then we also have an analysis and reporting system called ELISA, but that's a little more in the clinical space. And if you guys want more information, we can talk afterwards. We also offer a couple of products that are outside of that typical NGS workflow. So we offer a LIMS system called SLIMS, and that will track your samples from extraction or intake all the way through to your analysis and reporting. And then we also have automation options with our Bravo and our Magnus. The best part about this, coming from a former lab manager, is if you go with a workflow solution instead of 7 different vendors at a non-Agilent solution, you have one account manager that you're dealing with. You have one, if there's a problem, one of my coworkers jokingly calls it the one neck to throttle. So you have one person that you're contacting instead of up to 7 vendors with other companies. So starting to go through the NGS workflow and stopping at QC, our automated electrophoresis portfolio, we have a wide range of options for QC of samples, both before starting library prep, during the NGS workflow processing, and any other time that you just need to do quality assessment of DNA, RNA, or protein. As you can see from the slide, we have various throughput options. You could run 1 sample up to hundreds of samples processed at a time. There are various sensitivities and sample type options that can all help you select the instrument that's right for your process. I do want to highlight the 4150 and the 4200 tape station. Those are currently our best selling products. And I do also want to mention the femto pulse. There is a white paper that was released recently, if you're doing any long read sequencing, about using the femto pulse to do some of the QC for long read sequencing. So continuing, library prep is the next step. Agilent has a large variety of library prep options, as you can see from this chart, depending on what starting material you have, DNA or RNA. Each of these library prep solutions has some slightly different features. So we can actually pretty much tailor your library prep to whatever your lab needs. What's circled here is our newest library prep option that was released early 2020, actually right before I started at Agilent. And what this does is it actually combines some of the features of our other methods into one all-encompassing DNA, RNA, all sample types workflow. And we're going to talk in detail about that. So XTHS2 is a parallel process that you can use for the sequencing of DNA or RNA. It's the same library prep steps. If you're doing targeted RNA-seq, it's the same target enrichment steps. If you're doing RNA, there's a cDNA module that comes before, or if you're doing mRNA-seq, we have a poly-A selection module. You can buy pieces of this workflow, whatever you need. We tried to make it as flexible as we possibly could. It also uses our fast time, which means you can actually complete this workflow in one day. It is a long day, but you can do it in a day, or you can hold on a thermocycler overnight and start processing again the next day, whatever your workflow needs. Some of the other features that we're going to talk about in a little more detail include the 384 dual sample indexes and dual molecular barcodes. This chemistry has both of them. And all of these features lead to a workflow that gives high library complexity and low duplication rates, and is compatible with multiple sample types, including FFPE and cell-free DNA. So talking a little bit more about those 384 unique dual sample indexes and why they're important. Once multiplexing became a thing in sequencing, a phenomenon called index hopping was noticed. And that's where a misassignment occurs that results in the incorrect assignment of libraries from the expected index to a different index. With the introduction of patterned flow cells by Illumina for their HiC, and especially for the NovaSeq, that problem kind of compounded a little bit, and people were starting to see index hopping rates of around 5%. This can actually lead to an increase in false positives in your low frequency variants. If you use dual indexes, you're able to filter out those misassigned indexes, and therefore they're not going to affect your data. And with 384 unique indexes, you have the ability to maximize your throughput. These primers come pre-mixed. They either come in a strip tube or a plate. They're added in a single step. This is a change if you've used some of our older chemistries. It used to be a two-step addition and a little cumbersome. And also with our previous chemistries, you had to choose whether you wanted to do a barcode or an index. With this one, you get the ability to do both. So another feature of the XTHS2 chemistry is the option for molecular barcodes. Anytime you have amplification occurring, there is the potential for PCR duplicates to be created. PCR duplicates are duplicates that come from the same starting material. When fragmentation occurs, either by mechanical or enzymatic means, you also get duplicates created. These fragmentation duplicates are actually copies that start from different starting material and can provide you useful, unique information. Without molecular barcodes, all reads with a common start and stop sequence are assumed to be PCR duplicates and are removed. There's no way to differentiate between your fragmentation and your PCR duplicates. So that means that you're not only losing the PCR dupes that you want to lose, but you're also losing your fragmentation duplicates as well. Doing this means that you lose some of the unique read information that the fragmentation duplicates provide and can impact your reaction sensitivity. Molecular barcodes are unique sequences that are used to tag the starting fragments, and they're introduced during the adapter ligation step before amplification occurs. And this allows you to differentiate between your PCR and your fragmentation duplicates. Now, any duplicates that have the same molecular barcodes can be removed and leave your fragmentation duplicates alone. We actually can take that one step farther by using molecular barcodes to help build a single strand consensus sequence if you're using single molecular barcodes, or a duplex consensus sequence if you're using dual barcodes. The process for both of these is the same. You start by aligning the data to the reference genome. and then identifying all of your duplicate reads. You group those reads by the molecular barcode sequence. And as you can see from this cartoon illustration, in this barcode family, there are some random errors that were introduced during PCR, and then there's one true variant that's seen in all three fragments. All of the information in the barcode families are consolidated in a consensus sequence for that barcode. And this process actually helps you filter out your PCR errors from your true variants. And then taking it another step farther, if you have dual molecular barcodes, you can build a, you take the information from your plus strands and your minus strands, and then you combine that to make a duplex consensus sequence. And dual molecular barcodes are critical for any application which requires low variant allele frequency detection like liquid biopsy. And I do want to note that if you want to take full advantage of this dual molecular barcodes, you do need some pretty deep sequencing coverage. But even using single molecular barcodes shows a significant reduction in your false positive calls. In this graph, all of the cells highlighted at the bottom of the screen in the red box are actually calls that were removed using barcodes and allowed for higher specificity in the data. By using dual molecular barcodes, you get about a 74% reduction in false positive calls when you compare to using single molecular barcodes, and a 93% reduction when compared to using no molecular barcodes at all. So XTHS2 is a parallel workflow, and that's great for molecular barcodes for DNA, but it's also critically important for RNA-seq as well. As the three graphs on the screen show, the deduplication with molecular barcodes actually allows for accurate expression profiles for low input samples and high sequencing depth libraries. If there's no deduplication, the data leads to systematic overestimates, and using only chromosomal coordinates can lead to an underestimation of your high expressors. By using the deduplication of chromosomal coordinates with molecular barcodes, you get great data correlation. Another important feature of the XTHS2 chemistry is better conversion efficiency when compared to our competitors and when compared to our previous chemistries. For DNA, the better the conversion efficiency, the higher the DNA sequencing library complexity is. And conversion efficiency is just a measure of the percentage of input DNA molecules that are converted into fully adapter ligated libraries, or to put it more simply, how much you started with actually becomes a library. This high library complexity is critical for degraded or low input samples, and the ability to get high library complexity from this chemistry means that you don't have to use a special workflow or an adapted workflow for your degraded or low input samples, again, simplifying your workflow. The goal was to create one workflow for all. For RNA sequencing, better conversion efficiency means that you have better expression accuracy, and this will give you a better chance to detect your low expressors. Doing deduplication using dual molecular barcodes also helps lower the duplication rate and improves your library complexity, therefore giving you better conversion efficiency. So moving on with our NGS workflow, we come to target enrichment. When talking about target enrichment, Agilent actually created the market for target enrichment when we introduced the first exome enrichment kit in 2009. And to date, that exome kit is still the most widely cited exome product in literature. Since that time, Agilent has been pretty committed to improving and advancing its SureSelect platform and target enrichment products. So when I like to talk about our target enrichment products, we have three different categories. We have our catalog panels, and those are commercially available products. Agilent creates these panels, they validate them, and they're available for anyone to purchase. And they include products like our all-new V8 exome that we're going to talk about in just a minute. We also have custom panels, and custom panels are just that. They're custom created by you and fit your lab's needs for whatever project you're working on. And our third category are our community design panels. And this group is content that's developed by or in collaboration with experts in various research areas, including oncology research, infectious disease, model organism exomes, and epigenetic research. And actually, since the slide deck was approved, we have actually come out with another community design. It's called StrainSeq, and it's used for cannabis strain identification through sequencing. And if anybody is interested, I can find out more information about it, but it was released maybe a week ago. Before getting into some of our panels and talking about them more in depth, I did want to share with you an update that occurred in the summer of 2020 that was pretty significant. This update included changes to the way that we design, print, and recover our RNA baits, but we kept the actual RNA bait making the same. So you're still getting the same high quality product as usual. And this process was internally and externally now called surge. So if you ever hear anybody saying surge or pre-surge, this is what they're talking about. So what they did is they completely went through and they rehauled our design process. So now probe selection and placement is done by machine learning. Our probe printing is now targeting both the plus and the minus strands to minimize base-specific biases. Our probe recovery, they created a completely new process that helps to minimize AT and GC dropouts and produces higher yield. And again, the RNA bait making is still the same, so you're getting still the same quality baits. Any design that was created prior to summer of 2020 will still be the same and you can still reorder it. But any design or any tweaks that are made after that summer is using the new surge process. So I just wanted to talk about a little bit more about the probe selection and placement. Machine learning is actually a smarter and more intelligent way to design the probes. What we used to do is we used to take the target region and then we would tile the probes evenly across that area. And we've switched that to machine learning now. And we're able to tile tighter on smaller targets and we're actually able to minimize the design footprint. That doesn't mean that that target area has changed size. It just means the probes that we're using to cover that are in a smaller area. And now you can actually fit more samples onto a sequencer, not because the design is smaller, just because the footprint is smaller. And this machine learning actually helps improve some coverage uniformity and again, reduce the design footprint. So I like this picture. Our all new V8 Axiom was released in the summer of 2021, so just a couple of months ago. And it is our first commercial product that was launched using the new surge design process. So V8 Exome is not only one of the newest exomes on the market today, it has some of the most up-to-date content on the market. It's also the core of a cluster of products that can be customized for specific applications by adding already created content to the V8 Exome. So if you wanted to do detection of CNVs and chromosomal aneuploidies, you could add the one-seq content to the V8 core. If you needed to detect mitochondrial DNA mutations, we have mito content that can be added. And coming soon, this is March, so maybe not soon, coming summer of 2022, we're going to be releasing additional content for the detection of cancer biomarkers, also known as tumor mapping. We did just release 2 additional products, the V8 plus NCV or non-coding variants for clinical exome sequencing, and the V8 plus UTR or untranslated regions for biomarker discovery and translational research. And those 2 products were actually just released November, December of 2021. So V8 has excellent coverage in the RefSeq exons and genes and the ACMG59 genes, more coverage than our competitors. And I will say the V8 plus the NCV and plus UTR actually has coverage on ACMG74 genes. V8 has excellent coverage of rare disease genes in both exon and gene level. So these metrics on the graphs were determined by looking at a panel of 438 genes that were responsible for 448 childhood recessive diseases that were analyzed at both the exon and gene level. And V8 was found to provide more than 60% more coverage at the exon level than our competitors. So moving away from our catalog panels, Agilent has a great reputation for designing custom panels and designing custom content in general. To create your custom panel, all you do is you can upload your targeted regions or genes into our SureDesign software. This is for human designs only. If you have a non-human design or something more complex, or if you would just like a little help getting your design as perfect as we can, you can work with your local FAS. And then we also have something that's new in the last 2 years called our design testing service. And this is a service where customers work with a specialized group of scientists to design the content for their panels. That content is then created in a 16 reaction kit, sent to our labs in California. They will use HapMap samples to do the sequencing with the content and send the data back to you. And then you can review the data and see if there's any further optimization needed. They can help you evaluate some of that after the data as well. If anybody is looking to do clinical sequencing, this can actually help take a couple of weeks off of validating your clinical sequencing run because you'll actually have some validated data. So custom designs can be used for DNA and RNA targeted sequencing, as you all know. But one of the newer applications from the last 2 years is using it for something like high throughput chromatin capture, conformation capture, or HiC. So chromosomes adopt highly complex architectures that maintain a compact structure, yet still support dynamic cellular processes such as transcription, replication, and DNA damage repair. Our understanding of these processes is growing rapidly thanks to technology like HiC. HiC probes the three-dimensional structure of chromatin via proximity ligation followed by massively parallel sequencing. It's a highly powerful technology due to its ability to capture and reveal the unique topological characteristics of the genome. Identifying individual structures like TADs or topologically associating domains and combining this data with epigenetic data can provide insight on how chromosomal architecture can impact cellular processes, like the enhancer activation of a distal promoter, for example. A process like whole genome HiC requires millions to billions of reads. And while it can give you some super high resolution results, it can be very, very cost prohibitive. A more practical approach is to use target enrichment to reduce the number of sequencing reads and reduce your cost while allowing those targeted regions to be sequenced at a far greater depth and increase the resolution of those areas. Traditionally, Capture HiC has been a pretty cumbersome and labor-intensive approach. And we actually paired with a company called Arima and used our XTHS2 library prep and target enrichment along with their HiC library prep kits. And we were able to create a streamlined and simplified 3-day workflow. The targeted probes can either be custom designed with the help of Arima scientists, or Agilent offers, we'll call them 2 catalog panels. One that's uses the HG19 build and one that uses HG28. An app note was created in collaboration with Arima, and we were actually able to confirm using the XTHS2 Arima workflow that we got resolution of the targeted regions all the way down to 500 base pairs. We uncovered chromatin conformation and epigenetic features around those targeted genes. We were able to do cost-effective sub-kilobase resolutions, and we showed improved resolution actually aids in the analysis of gene regulation. As I said before, the true power of Capture HiC lies in the increased resolution possible by focusing your sequencing reads to your regions of interest and getting a concentrated sequencing depth. The 3 figures on the screen are showing data from the AppNote collaboration and shows the captured HiC data of the CVR gene and the K562 cell line been to progressively higher resolutions. Graph A is at 25 kilobases, B is at 5 kilobases, and C is all the way down at 500. As the resolution increases, the long-range chromatin contracts, become more refined, and allow for a more precise identification of the topological landmarks and enable finer scale observations. So this is further observed by the chromatin loop. That's what the blue arrow is pointing to. That's anchored at the promoter and in the intronic regions preceding exon 9. which is actually what the blue arrow is. Below the three pictures is actually a table that shows the estimated sequencing depths that you would need to get to those resolutions. And like I said, you need a lot of sequencing to be able to do this. So moving to our third category, our community designs. We have the Glasgow Precision Oncology Lab created the Glasgow cancer panels for us. And those are 3 fit for purpose cancer genome assays for solid and hematological cancers. We have epigenetics research designs for both mouse and rat methyl-seq, which enable global DNA methylation profiling in those 2 key animal models. And those panels were designed in collaboration with a group led by Dr. Richard Lee at Hopkins. And they target comprehensive sets of CPG islands, shores, shelves, and regulatory elements. For our model organism exome sequencing, we have the mouse all exon V2 based on the MM10 genome build from Dr. Ganda's lab. And this design was used to publish a study to better understand molecular pathogenesis and leukemic relapse. The rat all exon was created by Julie Foley at NIEHS. And that design was used to study genetic changes in chemically induced rat tumor cell lines. And those results were published in 2018 in BMC Genomics. And then finally, we have a canine all exon that was designed by Professor Murchison from the University of Cambridge. And she and her group used that exome sequencing to study the evolution and global expansion of a transmissible cancer lineage in dogs. And that data was published in Science in 2019, August of 2019. And then our final category is our infectious disease panels, which have been they've been a rather hit in the recent years. The design of these panels was led by Professor Judith Brower at the University College of London and her research group. So they found that due to the much smaller size of pathogen genomes, relative to the proportion of host genetic material in an extract, there's much more host versus pathogens. So to overcome that issue, we've actually created several different modifications to our standard short select protocols for enrichment and sequencing of these pathogen genomes. It's in an app note if anybody's interested, I can direct you to that. These genomes for these infectious disease panels were actually taken from various clinical samples from blood, sera, plasma, stool, urine, sputum, and nasopharyngeal aspirates. The probe libraries were designed to capture the whole genome of the pathogen. And these pathogens include RNA viruses, DNA viruses, and bacteria. And then these libraries are based on comprehensive sequencing database for each organism, and again, allow you to get deep sequencing for the entire pathogen. So continuing to move along on our NGS pathway, we come to automation. So we have 2 options available for automation. In the lab, automation can actually increase consistency across samples and users. It helps reduce error rates and allows more samples to be processed per day and can help freeing up some lab tech time. The Bravo is our high throughput option, and it's not just for NGS protocols. In fact, the Bravo has kind of had its heyday for qPCR setups for COVID. You can use this instrument to do qPCR assays to fill plates. You can use it in library screens, pretty much in any application where you want to move liquid from a reservoir to a plate, from a reservoir and a plate. Our newest automation option is our Magnus, and this is a completely walk away NGS automation option. It runs 8 reactions at a time. I have set this up. It truly does take 5 minutes of hands-on time to set the instrument up after your reagents have thawed. And it will take your sample it'll do enzymatic fragmentation of your sample, and then 9 hours later, you'll come back and you'll get your libraries ready to pull and put on your sequencer. All right, switching gears, we're going to move on to what we are currently calling our synthetic biology portfolio, which includes our DNA oligo pools and our single guide RNAs, along with some other CRISPR products. And we are getting ready to release some different CRISPR products in the coming year. So there is no doubt that CRISPR-Cas genome engineering has transformed the life sciences due to its simplicity and robustness. Agilent is invested in both ends of the spectrum with tools and technology used to quantify changes in genomic DNA and the platforms used to measure the impact of altering gene function. These edits and edited cells can be analyzed using a variety of instrumentation, including Agilent Seahorse for metabolics, our LCMS chemistry for protein analysis, and sequencing technologies for RNA function and off-target analysis. So I want to talk a little bit about how CRISPR works. In essence, CRISPR is like a scalpel for DNA that can be programmed to create a break in the DNA at a specific sequence in the genome. CRISPR has two main components, a protein, most typically Cas9, and a guide RNA that complexes with that enzyme and mediates its sequence-specific binding to a site in the genome that corresponds to a 20 nucleotide region of that RNA. Once the CRISPR complex binds to the correct sequence, the Cas9 enzyme comes and cuts the DNA. Once the break is made, repair enzymes in the cell come to fuse those DNA ends back together at the break point. And often this fusion is not perfect, and there will be either a small deletion or insertion, which can lead to that gene actually losing its function or being knocked out. You can also add new sequences by including a small piece of DNA called a donor DNA. One of the primary reasons that CRISPR is so much easier to use than any previous gene editing systems is to change your target. All you have to do is create a new guide RNA. I make it sound like that's nothing, but when you compare it to changing a protein, it is that simple. To change the sequence, you just design a new guide RNA. And as some of you already know, Agilent is a leader in DNA and RNA synthesis. So getting a little bit more in detail about the breaks that actually occur. The cell uses one of two primary repair pathways to heal that break. And those pathways are either non-homologous end joining or homology-directed repair. In non-homologous end joining, on the top right, you can cause insertions due to polymerases in the cell, adding some nucleotides to the broken ends and sealing those ends back together, or you can get deletions due to exonucleases in the cell, chewing back a few base pairs before those ends are ligated together. The ends are simply glued back together. There is no error checking. Non-homologous end joining is inherently a mutagenic process. For homology-directed repair, on the bottom right, you take the same guide RNA plus your enzyme like Cas9 plus a third component. You have a donor DNA template, such as a single-stranded oligo or a double-stranded plasmid donor, which is shown in the green in this picture. In this case, you get cleavage by the CRISPR-Cas, and then the cell uses homology-directed repair. They use the donor templates to do the repair. In nature, the cell would actually use the sister chromatid for high-fidelity repair, and we're actually tricking the cell into thinking that the supplied donor template is the sister chromatid, so you get directional synthesis and the copying of the new DNA sandwiched in between the homologous donor arms. So building a little bit on that classical gene editing workflow, there were two novel methods of editing genes that were introduced in the last couple of years by Dr. David Liu's lab. And those are base editing and prime editing. And these methods allow for targeted edits without a double-stranded break. Base editing uses a modified version of the Cas9 enzyme that only cuts one strand of the DNA that's referred to as a nickase. This nickase is refused to a particular deaminase, and this allows the complex to cut one strand of the target DNA, and then the deaminase removes the amine of a targeted base to convert a C to a T or a G to an A, and those are cytosine base editors. Another version of this uses adenine base editors to convert A to G and T to C. However, there are two issues with base editing. First, those are the only changes that you can make. And the second is this process is going to affect all of the nucleotides in that targeted region. So if your target region has more than one G, all of them are going to get converted to A's, not just the SNP that you're interested in correcting. As a result, you're fairly limited in how you can apply base editing. So the second method is prime editing, and prime editing also involves the use of a nickase, but this time it's fused to a reverse transcriptase. In addition, the guide RNA has also been modified, and it now contains a donor DNA template with the desired edit included, and this is referred to as a PEG RNA. The nickase cuts the targeted strand, but instead of using the host template to repair the break, the fused reverse transcriptase uses the DNA template provided by the PEG RNA. The original DNA flap is cleaved off and naturally excised by the cell, and the resulting mismatched double-stranded DNA with one correctly edited strand and one unedited strand is further resolved by introducing a second nick to the non-edited strand, thereby coaxing the cell to remake that strand using the edited DNA as the template. The result is you get a fully edited double-stranded DNA with the edit now present in both DNA strands. And this is an extremely powerful new approach that allows researchers to add DNA or edit DNA that's already existing in an extremely efficient manner. Insertions, deletions, replacements, or a mixture of these can be accomplished with prime editing. And an oversimplistic way to think of all of this is to think of your CRISPR-Cas systems as analogous to a pair of scissors, base editing as like a pencil, and your prime editing as like using a computer. So Agilent is a world leader in oligonucleotide synthesis. Both DNA oligos from our microarray platform and our RNA oligo synthesis. Agilent oligo microarrays and oligo libraries are used in many products for research customers and are also key components in technology used in clinical diagnostics. We have a separate division located in Colorado that does our contract manufacturing of our oligonucleotide-based therapeutics. When CRISPR exploded onto the scene, you can bet that we definitely took notice that a key component is actually that RNA nucleotide. About a year before the CRISPR craze started, researchers at Agilent Labs published a paper describing a new method for RNA synthesis, tcRNA chemistry. Although synthesizing a new RNA is a lot easier than making a new protein, traditional methods of RNA synthesis are good at making relatively short RNAs, whereas the most popular guide RNA size is about 100 nucleotides long, which is actually pretty long for a synthetic RNA oligo. The researchers that developed the tcRNA synthesis chemistry were motivated by new scientific discoveries about the many important roles RNA plays in human and other organisms. To study how the RNA works, researchers needed a way to get high quality, longer RNA oligos than traditional methods are able to make robustly. Again, we kind of hit the market right at the right time and had a proprietary chemistry that gives us the ability to make some of these pretty long and robust RNA oligos that are perfect for CRISPR work. Directly synthesizing your RNA oligos by chemical synthesis has some distinct advantages. First, you can incorporate chemical modifications into your RNA during this chemical synthesis. You can also make RNA in a pretty scalable way. One of the advantages of our tcRNA chemistry is that it works well on synthesizers used to make small amounts of RNA oligos, such as would be used for a research application, as well as the synthesizers that are used to make much larger amounts that might be used, might be created at our Colorado facilities in the GMP environment used for therapeutic applications. Although CRISPR is an incredibly powerful system, there are ways in which it can be improved, and we've focused some of our efforts on ways to improve our guide RNA performance by exploring some of the different modifications that can be incorporated during the chemical synthesis. The first area that we explored was RNA stability. RNA is still a rather fragile molecule. There are lots of enzymes in the cell that their only job is to chew up RNA. So the first modification that we created to our guide RNAs is at the end, shown in this picture. We added modifications to help stabilize the guide RNA. So where, if they get into a situation where they might typically be chewed up too fast to work efficiently, these modifications help mitigate some of that. You can also add modifications to RNA oligos that facilitate cross-linking to other molecules. like proteins or other nucleic acids you might want to complex your RNA oligo with. So again, there are several advantages to using chemically synthesized and modified single guide RNAs. Our high fidelity synthesis and high quality purification methods equal less off target and more efficient guides. And we are actually about to release a new purification process that actually allows us to purify a lot quicker and still get the same results. So you end up getting your guides a lot faster. Our chemical modifications help improve on-targeted editing, along with improving stability and result in less failed experiments. Our chemically synthesized guides are more stable, have a higher consistency and better quality over guides produced other ways. And all of this means the high performance guides from Agilent result in fewer failed experiments and difficult to work with cell lines. Our Sure Guide products, which is what they are called, are non-GMP, ultra high quality RNA that you can get in 100 micrograms to 100 milligrams. Anything over that has to go to our Colorado facility. And if anybody is looking to produce anything over that, I suggest you get on their list now because they are booked until 2024. They're building another one just to help with that. And we can do custom sequences up to 164 nucleotides long. We offer a variety of chemical modifications that are pretty standard, but we're also willing to discuss custom modifications upon request. And if you have a guide that you're looking at that you want that's more than 164 nucleotides, it's still worth a conversation. The more that we tell them that people want longer guides, the more they'll move it out of the GMP facility and let research have some of that as well. All right. So tying this back to sequencing just a little bit, this is a study that came out of CRISPR Therapeutics in Boston. And this is the use of hybrid capture sequencing to evaluate off-target sites, and it's becoming more prevalent. So this group evaluated 3 different types of sequencing: guide-seq, circle-seq, and site-seq, as methods for nominating potential off-target candidates of guide RNAs. They also used some computationally derived off-targets as well. They then used our SureSelect hybrid chemistry to capture not only the computationally derived off-target sites, but also the off-target sites derived from these 3 methods to analyze the precision of each assay. So just a little bit more about what those 3 sequencing techniques are. There is something called guide-seq, and that incorporates a double-stranded oligodeoxynucleotide at double-stranded breaks. And the primer sequences in that double-stranded oligodeoxynucleotide allow for identification of insertion sites and the nomination of off-target effects. Circle-seq is where we take genomic DNA that is circularized and cleaved via a CRISPR-Cas reaction. That linearized DNA is then sequenced to identify breakpoints and nominate off-targets. And then site-seq is where double-stranded breaks are enriched using biotinylated adapters ligated to a adenylated cleaved purified genomic DNA. And the off-target sites are nominated based on analysis of sequencing read pileups. So what the group at CRISPR Therapeutics found was that guide-seq was the most efficient and comprehensive assay tested. Circle-seq was also found to be useful in situations where guide-seq could not be used. And poor site-seq had some issues with correlation between read counts and indel frequencies. The study made several recommendations, including the use of both computationally derived on-target and off-target sites, along with sites derived empirically through guide-seq. The study also found that sequencing at a depth of at least 5,000x using hybrid capture was necessary for the greater than 0.2% sensitivity of detection. In another more recent study from 2020, Frangul and et al. Used hybrid capture to assess off-target effects of a single guide RNA. Their candidate off-target regions were chosen through computational analysis, and they also use guide-seq as coordinates for designing a custom hybrid capture library for assessing off-target effects in samples undergoing gene editing. This method assayed 224 potential off-target sites and detected only on-target edits and detected no off-target edits. So this is just a recap of why using SureSelect for your off-target analysis as some of the advantages that we have. Back to the dual unique molecular barcodes that allows for the detection of low variant allele frequencies. You can mitigate your index hopping and decrease your background and improve your base calling. And you got less false positive calls and more accuracy in your data. So the last thing that I want to talk to you about, and I promise I am almost done talking, is our short print technology. And this is generating high quality oligonucleotide libraries. And that starts with I cannot talk today, I apologize. Synthesizing high complexity pools of custom oligonucleotides. Agilent uses an array-based synthesis based on inkjet technology. There is a lot of backstory. Agilent actually spun out of Hewlett-Packard. So we can talk about that more after I'm done talking to everyone if you're interested. What happens is we deposit individual bases into defined positions on a glass slide. Those oligonucleotides are built up one at a time until the desired length is reached. And then these constructs are cleaved from the slide, pulled, dried, and shipped to the customer in a single tube. Our oligo libraries are then resuspended, amplified, and you can use them in any downstream application. So as kind of a repeat of what makes all of our products unique in the market, we have high fidelity customization and uniformity in our OLS products as well. Our OLS libraries have high fidelity, or they have more correct constructs and fewer errors per kilobase when compared with our competitors. And this means more clones are actually the sequence that you designed. Our higher uniformity means fewer misguides. And when discussing uniformity, a 90-10 ratio measurement is used. And that's a ratio of oligos represented at the 90th versus the 10th percentile. The closer that number is to 1, the better your uniformity is. Agilent has a 90-10 ratio of 3 compared to our competitors who range from 6 to 12. Having good uniformity in your OLS library will help find the hits in your screen instead of just the overrepresented ones, which translates to less screening to saturate your assay. And actually, we're getting even better uniformity. We are about to release a high fidelity OLS products that take that 90-10 ratio from 3 to, I think they said 1.5. And that those high fidelity oligos are coming out in a month or so. And finally, Agilent has the ability to customize and allow customers the ability to define the content of the OLS libraries. So you can determine the parameters of the experiment and you're not relegated to a whole genome screening approach when not needed. And it decreases your time and resources to get your results. The applications that these libraries can be used in are diverse. Some of those applications include massively parallel reporter assays, DNA digital storage, protein, enzyme, antibody engineering, genome and cell engineering, or CRISPR screening. We're going to talk about CRISPR screening in just a tiny bit more detail. Using OLS products to do a CRISPR screen is where you take a pool of guides, introduce them into a cell, and then you screen for your phenotypes. The primary workflow for any screening assay is to design your library. Then you amplify your library, introduce it into a vector or cassette via cloning. Then a phenotypic or functional screen is done to find out which cells contain your target ID. When doing a CRISPR screen, your library contains all of the guide RNAs that you want to screen for a particular phenotype. And this allows you to systematically find phenotypes associated with the gene or gene combinations that you want. Looking at a pooled functional screen library workflow in a little more detail, your pooled libraries start with a design of guide RNA panels. And this can be a genome-wide panel with more than 1,000 guides, or a more targeted approach like a pathway-specific panel with a couple hundred or a couple thousand guides. The guides are synthesized by our inkjet DNA printing technology. All of the oligos are printed simultaneously, and we can do up to 244,000 oligos at the same time, and those oligos can be in varying lengths between 30 and 230 nucleotides long. In mammalian cells, the guides are typically delivered by viral transduction, which means the DNA oligos are PCR amplified and cloned into lentiviral vectors, followed by packaging into viral particles. Once those particles are generated, they're introduced to a pool of cells at a concentration of 1 guide per cell. You do your screen, and the guides can either be enriched or depleted. Those enriched or depleted cells are then deep sequenced for those guides, and the hits are determined by the abundance of guide construct, which correlates to modification of a specific gene. For example, a readout of a CRISPR AI screen may also involve transcriptional profiling via RNA sequencing to identify the impacts of increased or reduced expression on pathways, which you can see in the yellow example. This slide just shows some different modes of CRISPR screening, and including your knockouts, your CRISPR-A activation, which activates your gene expression, and your CRISPR-I, which inhibits, represses, or dials down your gene expression. And then bringing it back to sequencing once again, the use of RNA-seq to assess transcriptomic effects of your CRISPR-AI. In this particular study, they actually use one of our older RNA-seq methods. Our SureSelect strand-specific RNA library kit was used to look at the gene expression changes arising from our CRISPR-A screen. But I do want to point out once again that SureSelect XTHS2 RNA can also be used to assess those transcriptomic effects of your CRISPR-AI screens. It's a streamlined mRNA sequencing approach. Also, if you're already using XTHS2 chemistry for anything else, it's the same library prep steps. You don't have to learn much different. You just have to, the polyA selection module or the cDNA module. Again, we have the dual molecular barcodes, and we've already talked about why those are very important in RNA sequencing. And this is it. We've come to the end of my talk. We have just discussed a couple of Agilent's technologies for CRISPR-Cas genome engineering, our synthetic RNA created via our TC chemistry, and our inkjet printed DNA for various applications, including CRISPR libraries. As many know Agilent for its world-leading analytical tools, such as LCMS, Agilent is also committed to bringing the same high-quality products into the genomic and molecular biology spaces. I want to thank you guys for listening to me for the last almost hour and learning about all of the updates from the last 2 years. I know it was a lot of information jam-packed into 1 hour. I hope everybody learned something, and I am absolutely happy to answer any questions that you want. Again, there is still a lot of water back there, so please hydrate before you leave. And I will also be back in this room at 11 o'clock tomorrow for a vendor show. Thank you guys. Are there any questions? Take a drink of water. What do you guys think? I'm also happy to answer individual questions if you guys want.
So you mentioned before use the barcode and barcode. How does that?
It depends. So it's deeper than normal sequencing, but it's going to depend on how big your targeted area actually is. It's pretty deep. I can get an exact number for you. You're not going to do it for exome sequencing, but some of the smaller targeted panels, it comes in very, very useful.
Yeah, my question, if I want to synthesize a long DNA, how many nucleotide can we synthesize in the DNA synthesizer?
So how many DNA synthesizers or RNA?
How many nucleotide in one DNA?
So for our DNA Oligo Libraries, you can do a pool up to 244,000 nucleotides. 244,000. Yep. And those can be varying lengths. So they can be 30 to 230 nucleotides long. You can do smaller lengths as well. You can do, I think our smallest category is 1,000. Don't quote me on that. I can look on the. I know the upper numbers. Nobody's actually ever asked me about the lower numbers.
Usually we want order some DNA from such as the IDT.
Mhm.
The website they pass through that is 100. 100 nucleotide. Base contain DNA contain 100 base.
Mhm.
If I want a longer DNA, then maybe I make another open this. But maybe I should need contact with.
Yeah, so we can, I know we can do a little bit more customization than IDT can. So we can, I mean, Agilent, the backstory of Agilent is we actually spun out of Hewlett Packard. So our background is a printing company. So whether it's the printing of ink on paper or printing DNA nucleotides on a glass slide, it's kind of our niche. So we can do some more of the complex. You're not limited to, so if you decide to create, 244,000 nucleotide library, they don't all have to be the same lengths. They can be of varying lengths. It doesn't have to just be human. It could be whatever organism you would like. And we did update, I forgot to mention this, another update that we had for our Oligo Libraries is it's now part of our Shore Design software. So you can create your own account and you can actually upload your own designs if you're comfortable doing so. We're still here to help you, but before it used to be you'd have to send me an Excel file and then we'd go back and forth about the Excel file rules. And this just simplifies it a little bit more. Any other questions? Thank you guys for listening to me talk. Thank you guys for having me in person. Definitely nicer here than it is in Baltimore.